these synthetic molecules can be a new class of medicine, of drugs.
Good morning, Victor, and welcome to Human Technopole. Thank you for joining us today, and we really appreciate you taking your time to answer our questions and sharing your insights with us. So, first question is that so you are among the pioneers of RNA interference, particularly small interfering RNA-based therapies. So, for the ones who are outside of the field, what is RNA interference and why is it considered as a major breakthrough compared to traditional medicines?
So thank you for your question. So every mammalian cells have special machinery which control gene expression on mRNA level. And this machinery, which scanning all mRNA in the cells, programmed by small interfering RNA. So what is interesting that this control is completely rational, and it's very quickly became clear that if we can do synthetic molecule as a small interfering RNA, these synthetic molecules can be a new class of medicine, of drugs. And because they can be designed to any gene in the body, they can be such universal solution for drug development. Plus, it can be completely rational. And number three, this molecule can be made by machine, by robots, by synthesizers. Mm-hmm. So it's very easy to make them. Mm-hmm. So it is why people very quickly realize that RNA interference is really breakthrough and can help to design a new class of drug.
You are the inventor of the drug inclisiran that works by switching off a gene and regulating the levels of cholesterol in the blood. So how does a discovery from the lab become a drug for patients?
So it's a dramatic story because so-called bad cholesterol dogma is one of the central dogma in medicine now. And everybody knows that bad cholesterol is really bad for our health, and it's major cause for heart attack and strokes, and it's major cause for human death, even exceeding maybe 70% of cancer death. So it's really very important to control cholesterol. And people from UT Southwestern in Dallas, they observe that there is a group of people which are very protected from heart attack and stroke. And all they have mutation in one specific gene. The name of this gene is PCSK9. And when you inactivate this gene, you have very nice level of cholesterol, very low level, and you will live much better life. So when I first heard about this gene, about PCSK9, I immediately recognized that if we can use siRNA as ideal tool to block transcription of this gene, we will have super potent drug, and everything is history. So we started this project. Unfortunately, we sold this project to another company, and in the end, this drug belongs now to pharmaceutical giant Novartis, and they're selling this in every country in the world.
Okay, so next question is, what is the strategic value of collaboration between academic research and the industry in advancing genetic medicine?
So it's a very good question. I strongly believe that there is really separation of role between academia and industry. So industry, according definition in biomedical area, cannot do fundamental discovery. They simply cannot do this because their mission is to develop the drugs. So academia, from other side, have another mission. They need to make discovery. And it is a very big distinction between academic research and industrial research. Nevertheless, both sides need to have dramatic collaboration. Only with such collaboration they can innovate, because academic people never can make drug development, which is very specialized type of biological or scientific activity. And the same for scientists in company. They are never under such pressure as academic scientists, which need to publish, they need to innovate. It's completely different culture of working. So it is basically ultimate goal for any good company. I strongly believe it's the case, especially in biotech. More collaboration with famous academic institution, better chances for success in industry.
So, looking ahead to the future of RNA interferon-based therapeutics, what do you see being the most successful application of these kind of drugs in the next 10 to 15 years? And what do you see to be the scientific and regulatory hurdles of such drugs in the future?
After 20 years work on RNA interference, basically there is no hurdles. There's only one hurdle, how to deliver this molecule inside the cells. If you know how to deliver, everything is super trivial, very trivial. It is rational design, all, basically regulatory and scientific hurdles is solved. Already at least 5 drugs are on the market. There is no problem with toxicity. They are super specific, super rational design. I see the future is super bright. Everything depends if people will know how to deliver this to different tissues. At present time, the whole progress was done for liver, but very close are solutions for muscle, for heart, and I strongly believe in next 5-10 years people will able to deliver siRNA in every cells in our body.
I see. So next question is, what responsibilities do scientists and researchers have to ensure that their innovation is not only effective but also accessible and ethically sustainable for society?
So it's very difficult question, because presumption from general public is that big pharma, big pharmaceutical company making exceeding benefit from discovery which made in the academia. And it is a wrong concept, because as professionals know, to make one drug it will take at least 7-10 years. Probability of success will be maybe 7-10% in the best scenario, and it will cost at least $1 billion. So when company taking such an incredible risk, plus investor taking such incredible risk, they basically assume that they will get profit from their investment, and this investment is super risky. So I have feeling that general public misunderstanding the goals and mechanism of sustainability of big pharma.
I see.
Great.
So that's the end of our question. Thank you for participating
Thank you very much.
and answering our questions. Thank you. So bye bye.